So one spent a couple days on methods, right? Talking about methods, thinking about methods, is because when we're in this class and other classes, we're going to talk about data collection. We're going to talk about statistics, right? And I'm not going to make you calculate statistics, but we're going to talk about numbers. I'm going to throw out a crazy number I just made up. 86% of people in America have tried, some illicit drug, right? I don't know. Just made up a number. You want to know, how did we get that number? Did we go ask everybody? Did we go ask everybody in America, and 86% of them said, "Yeah, I did some cocaine once"? No, that's not how we do these, because that's impossible, right? And it's impossible for a lot of reasons, particularly when we're thinking about drugs, right? There are some real problems with drugs, right? If we want to go, I'm thinking about developing a new treatment plan so that people no longer use heroin. Okay, so how are we going to do this? Well, let's just go grab a bunch of, if we're really doing this, if we really want to do this from a real pure scientific way, right, we're going to just go randomly grab a bunch of people and get them addicted to heroin. Right, that's step one. Okay, then step two, we have to assign a random assortment of them to the treatment group, where I'm actually going to do the thing that I think helps. And then the other group, I'm not, right? And so that's going to be, for some folks, ethically challenging, right? You can see the problems here, right? Okay, so then you think, okay, well, I can bypass this concern about giving people heroin. I give it to rats, right? There are some people who still have some ethical concerns about animal research, but not as many, right? And the concerns they do have aren't quite as powerful, and we're allowed to do this. We can give rats heroin. So then you think, well, I'm going to get a bunch of rats, get them addicted to heroin, and then I'm going to give them some kind of treatment plan. Now, the problem here, anybody have a pet rat? You have a pet rat?
I don't, but my girlfriend does.
Oh, okay. Well, that works, right? When she, you can do this with this rat.
No.
What can that rat do?
Kind of just eats and sleeps and poops.
Right. Which, and I understand if I asked you what do people do, they kind of do the same thing. But there's some more flexibility with people, right? There's some other things we could do. If I wanted to put a person into a standard sort of 12-step program, I may or may not recommend that, but let's say I'm going to. I can't put a rat into a 12-step program, right? If I send a rat to Alcoholics Anonymous, what's that going to do? Can you imagine? The rat's back there. I mean, they can't talk. You can't talk to them, right? So that severely limits the things you can do, right? You can do pharmacological interventions, certainly, right? We can give them a drug that might make them stop using drugs, right? While that might seem counterintuitive on some level, and we'll think about how that works later, you can do that, right? The problem is rat physiology is a bit different than human physiology, right? So there's some gap there, right? And so you see, whenever we try to approach this, whether we're asking people questions about their drug use habits, people aren't going to be forthcoming about that, right? How many of you, I don't know, they always gave us these surveys when I was in high school, right? They would ask you about two things everybody lies about: drug use and sexual activity, right? And so nobody, not necessarily together, but independently, they would always ask you about those things, right? Did they give you guys surveys like that, right?
Yeah.
How many of you were 100% honest on that? Okay, 2, right? The rest of you didn't raise your hands, and that's fine. I didn't expect even those 2. Because people lie on that, either one way or the other, right? Your friend's sitting next to you, and you're like, oh, absolutely, I've done that. That was a lot of fun. And you have no idea what it was. Look that up on the internet later. Or there are things you are doing, and you're like, I'm not supposed to do that. Do I want this guy to know I've been doing these things? I'm going to put no for all of it, right? And so and the data don't always wash out, right? So there's problems. But then there are also problems giving people drugs. So you have to look for ways sort of in between somewhere, right? Ways that are in between, ways that you can kind of take a whole series of studies that are using different methods, different approaches, and if they're all pointing in the same direction, then we can kind of learn something, right? Just because I give a rat a certain drug or do something to them and they stop using using a substance, that doesn't necessarily mean that's something that we can immediately just jump in and give to people, right? Okay. I know this really brilliant guy in Montana who one time, and you guys will read about this study. It's in the alcohol chapter. Who has alcohol? Not on them currently, but I'm in the chapter. Yeah, so there's this really brilliant study in there you guys should check out. There was this great group of folks. They used a really cool technique called optogenetics to make rats stop drinking alcohol. It was a brilliant research group. It was a lot of fun actually doing that. And so, well, you don't know what optogenetics is right now. Just imagine it requires me to put a laser in your brain. We'll just kind of, you can see where that might be problematic, right? Nobody's signing up like, I remember those little toy guns that had and you just have like a little laser thing there, and you're just like, oh, I don't think I want to drink today. I'm just going to pull the trigger, right? You can see where the applications can be. There's some steps, right? There's some disconnect there. So I think it's important for us to think about methods. I'm not expecting you to be an expert on any of these methods, right? When we're done, but I do want you to sort of have an appreciation for some of the problems that folks have when they're doing these studies. When you look at the data and you look at the information, I want you to be able to critically evaluate what other folks have done on some level, right? When it's coming time for you to do your presentations and you're doing research on a particular substance, if you can think about the research methods that they used for those sources you're going to give us, it's going to give you a leg up on what's going on, right? You're going to have a little better understanding. So I think over the next couple days, if we can take some time, talk about these methods, I think that's going to be great. The next Tuesday, we'll talk about sort of alcohol and drug use in general, like very addiction in very sort of broad terms. And then the following Thursday, what's that? You got it. That's the test. Which will cover the first 9 chapters. Okay. We'll work to get a study guide for you, right? Because I know that's like a lot of chapters, and there are a bunch of those we've skipped, like chapter 2 and chapters 4 through 8. We decided, right? 5 through 8. Right? I skipped chapter 4. Okay. Questions about that before we get in? Just kind of give you guys a roadmap where we're going. Does that work? Perfect. And Montana, I didn't check with you. Did you have something for today, or you think about Thursday, or not at all?
For optogenetics and kind of that.
Yeah.
Yeah, Thursday we'll talk about.
Perfect. So that's great. You get to hear somebody else talk for a while. What, 3-4 hours? He's got plenty of content. All right. So we'll talk about some research methods. We'll talk about a variety of different things that we can do to kind of look at the nervous system, right? There are about, I don't know, somewhere around 5 million different disciplines that are interested in how drugs work, right? And when I say how drugs work, that can mean how drugs work in terms of what do they make your brain do, what do they make your whole body do in terms of behavior, what do they make receptors and ion channels do, how do they so there are all of these different ways, right, Emily, that we can think about how drugs work, right? And depending on who you are and your background in your field, somebody comes up to you and go like, hey man, how's marijuana work? And then somebody's going to tell you, well, and then they're going to give you this whole long story about the biochemistry of cannabinoids and cannabinoid receptors, and you're going to go, I really just wanted to know, like, if I give this to my cat, is it going to get relaxed? And that's a whole different person you should ask, right? Okay. Sort of for our purposes, we're going to focus more on like behavior, mood, cognitive function kind of things. We'll briefly mention, and I hope you guys do mention and talk about some biochemical and molecular aspects, right? So some of those are important to think about, but we're going to think about that. We're going to focus more on this. And in particular, it's really important, as I said, for us to think about how do we quantify behavioral changes, right? So Montana, I know some folks working on a project where they're looking at aggressive behaviors, right? And so I know those folks have spent some time thinking about, well, how am I going to measure whether or not a mouse is more or less aggressive, right? Well, what are the specific behaviors I look for? How do I quantify that?
You'd have to find something that you can count, like a mouse doesn't really like shake its fist or get red in the face like a human would. So you have to kind of set an operational definition. Be like, we're going to define aggression as being when this mouse bites another mouse, when it stands up at this degree angle, it's demonstrating aggressive posture. the attack latency. How long does it take when you introduce these two mice for one to go after the other? Things like that you can objectively measure.
Which is not always the easiest. I know when I was in my undergrad, I for those of you that don't know, I used to give rats all kinds of drugs, and then measure their behavior, which was sort of interesting. But we were really interested in social interactions, and so one of the graduate students in the lab, he had to watch hours and hours of video and measure the angle that a rat turned its head toward another rat, and if it was like a threshold, it was like 25-degree angles or something, and if it turned its head more than 25-degree angles, that counted as a point, because that was a social interaction, you know? I know, right? So you have to think about those things. When we say you take a drug and it makes you less depressed, it makes you less anxious, it makes you, it gives you some subjective feeling. Like a lot of this is, for humans it's subjective, right? We can have you report stuff and we can try to quantify that somehow, right? That's something, but we also really like these objective benchmarks where we can do some math, right? All right. So again, we have to look at that, especially, when we make a change, whatever that manipulation is, how does that behavior affect, right? And we do want to compare that typically to a placebo, right? Placebo you can think of as like a control group, right? Okay. Sometimes when it comes to research, there will be multiple sort of control/placebo groups, right? So back in that example where we were trying to get people to stop using heroin, we might have the group who are using heroin, but we don't do any treatment. There's a group using heroin that we give them a classical treatment, the one that's previously been used, and then we have a group that they're using heroin, and we give them that new treatment that we're wanting to do, right? And then we can kind of compare, because again, one of the important things is if you come up with a new treatment, if you come up with a new drug, is it better than what's previously been produced? And if so, how much better? And does it have a better side effect profile? Right? That's kind of what you're typically looking for. All right. So again, we want to think about that neurochemical basis. A lot of this is going to come from animal models when we're thinking about sort of medications for psychiatric issues, right? You're not just going to go like, hey, I mixed together 4 chemicals. I think it's an antidepressant. Let's just get some people who aren't feeling so great and give it to them, see what happens, right? You're going to give it to a rat. How do you know if a rat's depressed or not? That's even more difficult than to know if a mouse is aggressive. You look for some other things. So there you go. And then before you even test this stuff, there's a whole group of folks who are like screening these drugs, right? Looking at these, looking at different things, and so you really want to sort of think about what you're doing before you do it. Now, if we're thinking about sort of animal research, right? What are the big advantages to animal research? Okay, one, we can definitely control living conditions, food, genetic background, right? The history of those particular animals, okay? Like what those animals have gone through, right? If I were to try to do a study, and I selected the folks in this room, and I would actually assume that the folks in this room are a tad more homogeneous than a random sample across the United States even, right? And the reason I say that is, how many of you are from West Virginia? Well, how many of you are not from West Virginia? Yeah, it's like 2 of us, right? Aren't we the great ones? But how far not from West Virginia are you?
Like an hour.
Okay, so that's really, unfortunately, not from West Virginia, right? So we're all from the same region. So we have similar sort of cultural backgrounds, right? How many of you are Marshall University students? Yeah, right. I know all of you. You don't have to raise your hands. So that again gives you some sort of similarity, right? At this point, things are heavily skewed in one sort of chromosome set than the other, right? And so, accepting, let's say, 3 of us, you guys have some of the same chromosomes, right? You're females, okay? And so that Brendan decided to look up at that point. You were one of the 3 exceptions. So, but females, right? So you have 2 X's instead of an X and a Y. And so, again, knock about 3 of us out of that. But you can start to think about maybe some genetic similarities, right? And some metabolic similarities. Males and females have different metabolism and they have different sort of enzymatic activity, right? So we can think about these things on some level. You guys are similar ages, right? Okay. I'm going to guess most of you are in that 20 to 22 range, right? I think it's going to catch most of you. A couple are probably below that. Again, Montana, we're already knocked out, so of this. But, even if I take the 10 or 12 of you who fall into that group, right, there's still a lot of differences, okay? There's still a lot of individual genetic differences. There's a lot of backgrounds. Some of you are from different socioeconomic statuses, right? Some of you are from single-parent homes. There's all kinds of different things going on there, right? That with a rat, I don't have to worry about. All rats have the same parents, right? All rats have the same, if you're using them for scientific purposes, they almost all have the exact same DNA. I mean, there's not really a huge difference, right? They're all nearly identical, which gives you some advantages and disadvantages depending on what you're trying to do. But even in that very narrow, in this very narrow group, I'm assuming most of you are psych majors, right? That's like another. Again, we're just right. Most of you, we're just adding layers of similarity, right? And there are still huge differences. I would still get big differences in your behavioral outcomes, even if I were to give all of you, who are almost as identical as I could make a group of people, right? Even if I were to give you, you guys would all have different outcomes if I give you the same drug, or do the same experimental manipulation. But with rats, I don't have that problem. They are all identical. They were all born at the exact same time. I can get, I can control their ages, right? I'm not worried about one of you who's a year older or 6 months older. I'm pulling the same rats from the same litter, right? I'll get 6 rats, and I'm in control of their life even before they're born, right? So that prenatal environment I can control. So there's a lot of things that you don't have control over in humans, even when you think you have such similar backgrounds. How many of you have a best friend who has almost the exact same sort of background as you, right? You guys maybe were in the same activities, right? You guys did the same things as younger folks. Your parents may even work at the same place, right? So you may have similar income situations, similar socioeconomic status situations, but you're still completely different individuals, right? If I had 2 rats and both their parents, worked at the hospital, they're going to behave exactly the same because they're rats, right? They're input-output machines. There's not a lot else going on there. The sort of additional advantage of an animal study is, again, that ethical concern, right? It's not typically ethical to give what we would consider an illicit drug, nor is it ethical and often, in the beginning, to give drugs for treatments of a disorder to humans, right? Most every drug that you take was first tested through rigorous, went through serious and rigorous animal testing. I say most every drug because I'm afraid there's like some weird exception of a drug that humans have been using for like 300 years, and the FDA didn't exist to make people do this, and so that's why I'm throwing out that one caveat for like that one thing, right? Like salicylic acid or something, right? That Neanderthals were chewing on. that didn't go through animal testing first before that guy just started chewing on it. But often things aren't ethical with humans, right? And so if we want to test a drug in the early phases of development, if we want to test an antidepressant, we need to test it, in another species first. If we want to look at some of the studies I was doing, we would give psychomotor stimulants to rats, and then we would give them dopamine receptor blockers. For either there are different kinds of dopamine receptors, D1, D2 receptor blockers, and we would see if those receptor blockers stopped their locomotor activity. For those of you that don't know, and I don't mean to ruin the presentation for those of you who are going to do cocaine for your presentation, it increases your physical activity, right? Okay, that's hopefully not a surprise to most folks. And it does the same in rats. If you give a rat cocaine, it moves around a lot more than a rat that doesn't have cocaine. But what we tried to do is we tried to look at receptor blockers, right? We block D1, we block D2 receptors. Does that control the psychomotor or the locomotor effects of that stimulant, right? And so that was some of the things we were looking at. So you can kind of tease these things apart. Not something you can do in humans, right? You don't want to get some humans, even if they're a human who's using cocaine, there might be, if they're an active cocaine user, there might be some ethical concerns when they do these studies on humans, when they give them drugs that try to prevent or mitigate the effects of illicit drugs, you have to identify folks who have been, users of that drug for some period of time, right? You don't just kind of randomly grab folks and go, hey, have you tried cocaine? I got a great study. I'll give you extra credit in class. Doesn't really work that way, right, Kaylee? You have to...
I'd take it.
I guess it depends on how difficult the class is, right? But even if we want to go, let's go back, let's dial this back to a less sort of... yeah, let's think about nicotine, right? Let's say I've come up with a new nicotine cessation intervention, right? Sounds great. So what do I want to do if I want to test this on humans? I don't want to go up to people who've never used nicotine like, you want a pack of cigarettes? Let's light them up. Let's see what happens. No, you want to identify folks who have been smokers for a number of years or have used other tobacco products, right? And then, try to go that way. So you can see sort of some of the concerns. Now, the nice thing is, non-human mammals and humans, our brains are similar enough, right? I mean, if you think about what were the list of activities that rat would do?
Eat, sleep, and food.
Yeah. How many of you do those things? You're not going to raise your hand for some reason or another, but I know all of you do that. Because if you don't eat, sleep, or poop, how many days you got before you're dead? Right? A few, right? And I just saw some of you a couple weeks ago. So I know in the last 2 weeks you guys have done those things, hopefully on a regular basis, right? All three on a regular basis. It's the secret to health. So for the things we're going to look at with these animal studies, we can make generalizations, right? It's going to be similar enough. And here's just a fun picture of a sad little kid. It's like the animals you don't see. It's the animals you don't see that really helped her recover. She doesn't look like she's fully recovered. She still looks upset about that, whatever the problem was. I mean, don't get me wrong. She looks.
Maybe they told her about all the animals they killed in the process to find those drugs.
And then she was like,
I like maybe
we
like,
lost some lab animals, but look what we saved. And she's like, I think they told her we killed some animals to save your life.
yeah,
this is because of you.
yeah. Actually, she had a malignant tumor, a brain, from her brain.
They told her, they're like, all these little rats had brain tumors, and we gave them to them. We gave them cancer, and that's why you're cancer-free now, because there were dead rats somewhere.
Either her brain's just messed up. There may just be something. Or maybe she just watched like a lot of America's Next Top Model and is trying to do that look that they all do.
Part of me wants to maybe watch that show so I know what you're talking about. No, I haven't. But thanks anyway. I appreciate it.
Really good. You should watch it.
All right. So let's talk about correlations a bit. You guys familiar with correlations? So this was something that maybe you don't think of as a correlational sort of situation now, but at some point it was, right? That's the issue. Like most everything that you know is causal at some point was a correlation that someone observed, right? And fetal alcohol syndrome is a great example, right? There were a bunch of folks who noticed, hey, people who drink tend to have kids who have a certain set of physical and developmental issues. Wonder if those two things are related. We don't really know, right? Because we didn't do a study. We're just like, hey, we've noticed this correlation, right? This correlation is here. So how did we demonstrate, how do we move that from correlation to a causal relationship? You do an animal study, right? You give some pregnant rats alcohol and then see what happens. And you give some pregnant rats, instead of alcohol, you give them a saline injection, right? You don't give them alcohol, you give them that control. And then you see what happens and you compare the outcomes. Then you know it's causal. You can go beyond that, and in fact, they have gone beyond that, and you can start to look at the specific sort of cellular mechanisms, right? How alcohol interferes with those developmental processes, how it interferes with the way that those structures are created. If you think about fetal alcohol syndrome, I think most of you are familiar with fetal alcohol syndrome and the sort of physical appearance of individuals who have been affected by that. But they also have some developmental issues as well. now we can take that correlational issue, we can turn that into a causal relationship, and with proper education and other sort of outreach, we can drastically reduce the cases of fetal alcohol syndrome, right? Can really kind of make that improvement. Now, despite the fact that people do animal research all the time, there are very strict animal care guidelines, right? There are a ton of organizations, federal government entities that have to make sure that you're doing things in the right way, right? When we talk about some historical research, we talk about things from some time ago, and you read some of these studies, some of the classic sort of things that folks have figured out, you think, wow, that's really kind of out there, right? Emily, we talked in the other class about Pierre Florenz, and sort of he just like scooped out parts of animal brains, and then he was like, oh, they can't breathe. That's the part that controls breathing. He was doing his work in the 1800s, okay? So it was a completely different situation, right? You're not going to get by with that now. And in fact, there are very strict regulations about how much space each animal needs, and if you need for some reason to conduct an experiment that violates those sort of basic rules, you have to justify that. You have to justify it quite a bit. Okay, there are a number of federal laws, right, that govern this humane treatment of animals. There's sort of, and you'll hear these words, and they're not synonymous. You'll hear these words sometimes. There's what we call animal welfare and animal rights. And while they sort of at some level sound the same, they're not. They're different. Animal rights, typically the word that goes next is activists. These are individuals who are advocating for no animals being used at all, right? I think the typical sort of group there is like PETA. You guys are probably familiar with PETA, right? I don't want to make a...
That's a whole can of worms. That's just...
No, they don't put worms in cans. Those are animals too. So be careful with your... And in fact, I think they actually have some problems with language like that even. Like they actually have problems with more than one way to skin a cat.
With the fact that they exterminate like 90% of the animals that they rescue.
Yeah. Have you played their Pokemon games? Where
No,
they
I
have
haven't.
Pokemon? It's hilarious.
PETA has Pokemon?
Yeah, like it's like Pokemon Black and Blue, and it's about how Pokemon like encourages animal abuse because you're taking them out of the wild and shoving them in these little balls.
They compare it to like dog fighting.
Yeah.
I'm like, it's a game. These animals aren't real.
And then they have Pokemon like Red, White, and Blue, but it's against McDonald's. What? Yeah, it's a lot of fun.
No comment on that one.
See, that's it. A whole can of worms.
It is. Wow. That's something. So to contrast that with animal welfare, right? These are individuals who see the value in animal research, but they recognize that these animals need to be treated appropriately and humanely, right? And so these, when you do these, I've indicated I've done a number of animal studies. We had to provide enrichment activities for these animals, rats, different kind of enrichments than other sort of species, right? But they do. There are a number of things that you have to do. You have to provide, make sure the diet is adequate, make sure they have access to, food and water, they have plenty of space, they can roam around. If it's a social species, you need to house them, with their buddies, right? So they can get along. If you, for some reason, want to do an experiment that requires you to not house them together, you have to justify that, and you have to justify that with a lot of scientific literature. So a lot of stuff you really want to try to reduce animal distress, okay? All right, there are folks here, most of you are probably more familiar with what they call the IRB, the Institutional Review Board. They probably spent a fair amount of time on that in your experimental psych class, for those of you that took that, right? There is a sort of similar board of folks who review animal experiments, right? We don't talk about those as much in this department. They're called the IACUC, that's the Institutional Animal Care and Use Committee. I know, it's not as fun of an acronym or series of letters, right? IRB, yeah, IACUC. It's like, it sounds a little worse, but anyway. So, That's what you have to do. You have to present things to them. They'll decide whether or not you are getting enough knowledge or benefit to society out of that. They'll typically ask you to figure out ways that you can do it that don't use animals, right? So there is a sliver of, people think about like all the animals that are used in research, and it's such a small number compared to some of the other sort of things that people don't get as much of an uproar. Now your Pokemon Black and Blue folks do, but if you think about like pets, for example, right, and the way pets are often treated, the number of pets and their treatment is greater and worse than typically what you think about for lab animals. Okay. And again, you're always trying to look to minimize discomfort, distress, and pain for a couple reasons. One is just the right ethical thing to do, and that's probably the biggest one. But also, scientifically speaking, you want to make sure you have robust, healthy animals, right, that are giving you good data. All right, animal tests. We use a concept called face validity. Do they closely resemble stuff on humans, right? So, for example, like blood pressure, that's an easy one, right? If you want to measure a blood pressure drug, the effects of a blood pressure drug, you can do that in an animal. Blood pressure kind of works the same in a rat as it does a human, right? Slightly different, maybe biochemical things going on. It's the same, right? The real problem is when you get into things that it's really hard to measure in animals, right? So, like, how guilty does a person feel after something happened, right? So maybe, what if I went over and I decided, like, hey, I'm going to take away 10 points from Laura's grade and I'm going to give it to Kaylee. And then I ask you, like, how guilty do each of you feel about that, right? I can get some quantifiable measure from humans because you guys can tell me that. If I go over to a rat and I take away its fun little grape it's going to eat and I give it to another rat, then it's hard for me to figure out how guilty the two rats feel, right? Now, there are, interestingly, a number of studies in non-human primates, right, in monkeys that have looked at cheating and detection of cheaters and how upset monkeys get over that. And in fact, there's a series of studies a while back where they gave monkeys will eat, they like grapes, but they'll also eat cucumber slices. But they love grapes even more than cucumber slices, right? And so they had two monkeys and they gave them the same job. And one monkey got cucumber slices and the other one got grapes, like equal number, right? And so think about this. The monkey who just got cucumber slices obviously got upset, right? Why did that monkey get grapes? But amazingly, the monkey who got grapes got upset too. Because because they realized, like, hey, my buddy over here got cheated. Yeah, I got my grapes. I'm not gonna give him grapes necessarily, but I'm gonna get upset about that, right? And so it's kind of interesting. Some of the things that you can look at with other species, in particular non-human primates, and they're quite fascinating. What's the problem with using non-human primates for like drug studies? They're expensive and they take up a lot of space, right? And most people, like that little girl we just saw, she would probably get more upset if we said we had to kill 50 monkeys than if we had to kill 50 rats, right? For some sort of odd reason, you can think about whatever that odd reason is, people have a hierarchy of animals, right? And whether you think that's founded in any sort of biological reality or not, that's the way it is, right? Most of you don't get too upset about rats. However, if I told you about studies that used dogs, I know some people are getting upset there, right? Actually, when I was in graduate school, there was a lab that was using beagles to study. Let me tell you. So they are the folks who keep you from getting HPV. So think those beagles.
Why specifically beagles?
Yeah, so beagles actually, interestingly, they get warts on their mouths and they just disappear on their own. They have a sort of like a natural process that takes care of it. So what they would do is they cut the warts off. Not so bad, right? I mean, if you got a wart, have it removed, right? And then they would take the warts off and then they would kind of study the biochemical process so that they could develop these vaccines. When they were done with the beagles, they gave them away. And so people got beagle puppies. There was always a waiting list. As you can, because they didn't need that many, right? Because these warts just sort of randomly pop up and you can have them removed. So, not such a bad story after I told you, right? Why beagles? Well, that's why beagles. And they're cute. So you can get rid of them easily.
Yeah, I guess that helps too.
Yeah, I mean, if they were ugly dogs, but I think it's the beagle breed in particular is more prone to these warts, and so you can collect more. It's very interesting. Yeah, so there you go, right? So, you know, were beagle warts worth an HPV vaccine? I don't know, probably, right? I mean, if you think about it, right? I mean, it's not such a bad trade, right? So there you go. So we need to typically find something measurable. Montana highlighted that when he talked about aggression. You know, if I give you a drug and I say, hey, you feel more or less aggressive, you can probably tell me that. The rat can't, the mouse can't. We have to measure that because we measure it in humans too. If we think about predictive validity, it's not a big deal. We're going to do a test. Hopefully, whatever happens in that experiment is what happens when we give you the drug or give the drug to the public. Okay? Not a big deal. If we're doing a study on animals, we want it to be predictive. It's going to be specific for that particular drug. And what's really important is this therapeutic range, right? So we want to have that nice dose-response curve, that nice dose-response relationship, right? If we have to, you know, flood an animal with some particular drug in order to get a response from it, that doesn't seem like probably a good idea, right? So we kind of want to have that nice sort of situation. We do want to make sure that tests are reliable. Okay. We want to keep doing the same thing over and over again and getting the same result. That's why human studies are problematic, right? I can do the same thing to each and every one of you and I'm going to get a different result. If I had a dozen rats and I did the same thing, I'm going to get the same result. If I don't get the same result, then what I'm doing is not doing anything, right? We can move on. All right. We have some different sort of things that we might measure with animals, some very simple things. I mean, are they having tremors? Do they salivate? How much are they moving around? Are they falling asleep? Right? These are all things that are pretty easy to measure. Now, one of the things that we might do is we might do what's called like operant conditioning. So we're going to have to train an animal to do something, right? This is particularly important. Like, let's say I was testing a drug and I thought it had some sort of cognitive effect, right? I thought it had some sort of effect on learning and memory. I thought it had an effect on attention span, right? And I give this to a rat. One, I need to drastically adjust my expectations, right? For rats, because their ability to learn and memorize is not your ability, and their ability to pay attention is a little different than your ability, right? But once I've adjusted my expectations, I may need to train that animal to do something. I may need to train that animal to select a certain response. I may need it to, remember that when I see this image and I press a lever, I get a reward. If I see this image and press a lever, I get a shock. How quickly do they learn that under these different medications, maybe, right? And so we really want to make sure that we think about that. Some important aspects about this are what we call the scheduled reinforcement. I don't want you to get too bogged down with this, right? This is more of like something you get from like a learning course. If you guys haven't taken a learning course and you're interested in this, I recommend it. But there are sort of different ratios. We think about fixed ratio or fixed interval. Typically we mean like, if you do something, do you get a reward every time? If you do something 3 times, do you get a reward? Like, what's that? How often do you have to do something to get the reward, right? We want to. Some of these are variable, but some of them are fixed. It's not too big of a deal. Here's a rat sticking its nose into a pellet dispenser. You can see it's got a little lever over there. When that light comes on, for example, it presses the lever and it gets a treat. So now it knows every time the lever or the light comes on, press the lever, get a treat. You can make that lever or that light different colors even, right? Maybe it's red, maybe it's blue, green, whatever. You can do different things. All right. What about if we wanted to measure pain in a rat, right? So what if we're testing a pain relieving medication, right? One of the big things they'll do is what's called a tail flick. Basically, you heat up the tail of a rat and see how long it takes before it moves it. You're not burning it, even though it shows it's red. But how many of you have ever put your hand close to a light bulb? It's warm. There's a certain point where you'd move your hand, right? I mean, it happens. And so that's what they're going to do. Let's say this is like the normal time it takes it to move, and then after it takes the drug, notice sort of how much longer it takes before that rat moves its tail, right? So that tells you it's had some effect, right? It's actually reduced pain, okay?
Does that the board that tells you what you can and can't do. But after a certain amount of time, you have to like move the mouse itself, or if it just burns.
It depends. Yes, yeah. So it would depend on what you were trying to do, what your outcomes, what information you were trying to get, and how you had created that. So all animal studies have something called adverse outcomes, right? This is when you say, here are the things that can go bad, and here's what we're going to do if that happens, or to prevent that from happening, right? Because there are unexpected things that happen, right? Happens all the time. How many of you, so like here's a common thing, like let's say you're going to do some sort of manipulation to an animal's brain. You have to cut the scalp open, you have to get in there, right? You have to worry about things like infections, the animal's removing the stitches, right? And so you would list that. You would list that in your adverse outcomes. You would say, hey, if this animal gets an infection, here are the humane ways we're going to handle that. If the animal pulls out its stitches, here are the humane ways we're going to handle that. Sometimes it could cause you a problem with the data you're collecting. Sometimes it doesn't. It depends on what you're doing and how you're doing it. But that would all be included in that protocol. And those are important things. Yeah, because they'll ask all the time. I mean, and that's infections are one of the big things. That would be something in this case. If you're, this was when I was in graduate school, we used a species where we had to. We couldn't buy them, so we had to breed our own, right? So we had to monitor. I don't know if you guys are aware, like sometimes when two animals, try to make more animals, one of them's more enthusiastic about that than the other, and some injuries can occur. And so we had to monitor that. And if there was an injury, we had a protocol. Here's what we're going to do to fix that. We're not going to put those two back together because for whatever reason they didn't get along, and so we would, come up with other pairings and so forth. So yeah, there are a ton of things. And you do this with humans too, right? I mean, you have the same thing. If you're doing a study with humans, you're going to have the same. If you're doing a drug study in particular, you're going to have like, oh, somebody took the drug and their tongue swelled. Let's give it to them again and see if that happens. I don't think that's how that works, right? And so you'd have that same sort of protocol in place, and you try to think about all of the ways it could go bad, and all of the things you're going to do to prevent it from going bad, and then if something does happen, how do you fix that in the most humane way possible? Yeah, the stitches coming out are common in situations like that, and there are different ways that, depending on the situation, how you fix that.
Okay.
Does that make sense? Mm-hmm. All within what the protocol says. If you violate that protocol, you can lose your ability to do any research at all. In fact, your entire institution could lose its ability to get federal funding. So there's a very
like there are incentives to follow these policies.
Yeah, very much so. And there's a lot of peer pressure because you can imagine researcher A over here doing his bit, doing it the right way, cavalier researcher B over here just shooting from the hip on something, screws everything up, and then Marshall no longer gets federal funding, right? Guess who's going to be upset about that? All the other folks who were doing everything right. So there's a lot of pressure to make sure you do things right. Now, that's not to say that sometimes things change, and you have to submit amendments, and there's a process for that, right? Because you get into the middle of a study, and you're like, oh, this isn't working. Put it on hold, do the background work, try to get a different sort of protocol in, a different change, and you can do that. Okay. So there are methods for that. And a place, so in general, most places are monitored by 3 and 4 different agencies. It sort of depends. It was particularly bad when I was, as a graduate student, the species we used, they're called tree shrews. They're kind of like small little squirrel things from Southeast Asia. It's not terribly important to you, except the USDA did not consider them small animals. They were considered large animals, even though they only weighed like 200 grams. So the issue there is, so we had the institutional folks who came in. My graduate school was approved by another sort of, there's a group called ALAC. You don't have to know what that stands for, but it's an extra animal lab approval that you can go through and go like, not only are we just like at the standard, we're better than the standard in taking care of our animals and doing what we do. The DEA can come in if you're using controlled substances, right? Any anesthetics, the DEA can randomly show up. Lots of fun there. The FDA and the USDA can all show up randomly at your doorstep and go, let's see what you're doing. And if you're not doing it right, you got to fix it. And if you're not doing it right so bad, you're done. So, Yeah. So there's a lot, and we had when I was in graduate school, we had the DEA come out. We had reviews constantly from the internal folks, our external, the ALAC folks who came in, the FDA, the USDA were there quite regularly as well. Just making sure we were doing things. So what they'll do is like the FDA will assign somebody, or the USDA will assign somebody to a certain region, and they'll rotate around, and they'll go and they'll visit all the facilities in there. And we gave them fits because most of them were used to cows and pigs. And they would come in and they're like, what's this thing? And I'm like, well, it's a large animal.
Yeah, I just Googled that and it said that like the common tree shrew is like just over 7 inches and the pintail tree shrew is like 5 inches.
Yeah, so
how is that a large animal?
Yeah, because it's not a rodent. So for research purposes, anything that's not a rodent is probably a large animal. So when I went to graduate school, we had cows and we had pigs. There were folks doing like cardiac research. We also we got woodchucks. Somebody was looking at like hibernation. You know, like that's kind of interesting, right? We had the tree shrews, and then of course there were folks who had mice and rats as well. I think they had ducks there for a while before I got there. I know, right? Ducks. Once ducks. Yeah, so there you go. I can tell you all kinds of interesting things about tree shrews if you really want to know. They're fascinating little animals. All right, let's stop there for today because it's like the end.